Have you ever wondered how scientists separate mixtures in the lab? Let's explore the fascinating world of chromatography, a technique used to dissect complex mixtures into their individual components. Chromatography, a term derived from the Greek words chroma, meaning color, and graphene, meaning to write, is a staple in scientific research and industry. It's a bit like a race where different components of a mixture compete against each other. But instead of running on a track, they move through a stationary phase influenced by a mobile phase. Picture this, a bustling city during rush hour. The stationary phase is akin to the city's network of roads, while the mobile phase can be thought of as the stream of vehicles navigating these roads. Each component in the mixture, like different types of vehicles, moves at its own unique speed. Some rush ahead, while others lag behind. This differential speed of movement, based on the differing chemical properties of each component, is what allows for their separation. In the vast realm of chromatography, different types exist, each with its own unique method of separation. These include gas chromatography, liquid chromatography, and the one we're focusing on today, paper chromatography. Paper chromatography, as the name suggests, involves the use of paper as the stationary phase. It's a relatively simple yet powerful technique. Here, a drop of the mixture is placed on the paper, and the mobile phase, usually a solvent, wicks up the paper by capillary action, carrying the components of the mixture along with it. As the solvent moves, the components of the mixture separate based on their different rates of travel. Over the years, the methods of separation used in chromatography have evolved, making it an indispensable tool in scientific research. Among these, paper chromatography stands out for its simplicity and effectiveness. It's like the unsung hero of the lab, quietly doing its job and providing invaluable data to researchers. The methods of separation used in chromatography have evolved over the years, with one of the most effective being paper chromatography. The journey of paper chromatography began in the early 20th century. Like many scientific advancements, the story of paper chromatography starts with a spark of curiosity and a keen mind ready to explore. That mind belonged to German chemist Fritz Preyer, who in 1944 pioneered the initial development of this technique. Preyer's work was a game changer. He discovered that when a drop of colored liquid is placed on a piece of paper, as the liquid spreads, it separates into its constituent colors. This simple but powerful realization opened up a whole new world of possibilities for chemical analysis and identification. But the journey didn't stop there. Enter Archer John Porter Martin and Richard Lawrence Millington Synge, two British chemists, who saw the potential in Preyer's work and took it to the next level. In the late 40s, Martin and Synge refined the process of paper chromatography, making it more precise, reliable, and applicable to a wider range of substances. The duo's development of partition chromatography was revolutionary. They introduced the concept of stationary and mobile phases in chromatography, which allowed for the separation of complex mixtures with unprecedented accuracy. For their groundbreaking work, Martin and Synge were awarded the Nobel Prize in Chemistry in 1952. Their achievement was not just a personal triumph, but a significant leap for the scientific community. It was a testament to the power of curiosity, collaboration, and relentless pursuit of knowledge. It's fascinating to think that what started as a simple observation of color separation on paper has evolved into a sophisticated technique that scientists and researchers across the globe use today. From the labs of universities to the research and development departments of major corporations, the influence of paper chromatography is far-reaching. The work of these pioneers laid the foundation for the modern techniques of paper chromatography that we use today. Without the contributions of Fritz Preyer, Archer Martin, and Richard Synge, our understanding of chemical compounds and our ability to analyze them would be significantly less advanced. Their work has truly stood the test of time and continues to shape the world of science. So, how does paper chromatography work? Well, let's take it step by step. The process begins with a strip of high-quality paper, often cellulose. This paper acts as the stationary phase in our experiment. You see, the stationary phase is where the magic happens. It's the stage where our mixture will separate into its individual components. Next, we mark a line near the bottom of the paper strip and apply a small drop of the mixture we want to separate onto this line. It's crucial to make sure the drop is as small and concentrated as possible to ensure a clear separation. After the sample is applied, the paper strip is then placed in a container with a small amount of solvent. This solvent, or the liquid phase, is carefully chosen based on the specific mixture we're analyzing. The bottom of the paper strip touches the solvent, but our sample line does not. The solvent begins to ascend the paper by capillary action, meeting the sample on its journey upwards. As the liquid phase moves up the paper, it carries the components of the mixture with it. However, not all components travel at the same speed. Some substances have a greater affinity for the paper, while others prefer the solvent. This difference in attraction causes the substances to move at different rates, leading to their separation on the paper. Once the solvent has almost reached the top of the paper, we remove the strip and allow it to dry. What we're left with is a series of spots or bands along the paper strip, each representing a different component of our original mixture. We can then identify these substances based on how far they've traveled. And there you have it. The beauty of paper chromatography lies in its simplicity and effectiveness. Through a straightforward process, we're able to separate and identify the components of a complex mixture, all with a piece of paper and a bit of solvent. The technique of paper chromatography can be adapted in several ways, depending on the specific needs of the experiment. This flexibility is largely due to the variety of chromatographic papers available, each offering unique properties that can significantly impact the outcome of an experiment. First up is cellulose paper. As the name suggests, this paper is made primarily from cellulose, the most abundant organic polymer on Earth. In the world of paper chromatography, cellulose paper is the equivalent of a reliable workhorse. Its hydrophilic nature allows it to absorb a wide range of solvents, making it a versatile choice for many applications. The water molecules in the cellulose create a network of hydrogen bonds, which can interact with the molecules being separated, influencing their migration rates. Next, we have ion exchange paper. This type of paper is impregnated with ionic substances to allow for the separation of ions and polar molecules. It's an excellent choice when working with compounds that have a charge, as the ionic substances can attract or repel the compounds depending on their charges. This attraction or repulsion can significantly affect the migration of the compounds on the paper, enabling the separation of compounds that might be difficult to separate on regular cellulose paper. There's also the option of chemically modified paper, where the paper is treated with a specific chemical to enhance its affinity for certain types of compounds. This modification allows the paper to selectively absorb certain compounds over others, providing a higher degree of control over the separation process. Finally, let's not forget about thin layer chromatography (TLC) plates. Although not technically a type of paper, TLC plates are often used in similar ways to chromatographic paper. They consist of a thin layer of adsorbent material such as silica gel, alumina, or cellulose deposited on a flat, inert substrate. TLC plates offer higher sensitivity and resolution than traditional chromatographic paper, making them a popular choice for many researchers. Selecting the right chromatographic paper is crucial for achieving the desired separation. Understanding the unique properties and capabilities of each type of paper can help you tailor your paper chromatography experiments to your specific needs, ensuring you get the most accurate and reliable results possible. The versatility of paper chromatography has made it a crucial tool in various fields. Its uses are as diverse as the sectors that employ it, from food industry to forensic labs, from environmental studies to pharmaceuticals and beyond. Take the food industry, for instance. Ever wondered how food manufacturers ensure the consistency of color in their products? Well, paper chromatography plays a key role here. It's used to analyze food dyes and ensure they meet the necessary standards. This not only helps maintain the visual appeal of the food items, but also their safety. Now let's talk about the intriguing world of forensic science. In crime scene investigations, paper chromatography can serve as an invaluable ally. Be it identifying substances found at a crime scene or analyzing inks used in forgery cases, this technique comes into play. It allows forensic scientists to separate and identify complex mixtures, providing clues that can help crack a case. Moving on to environmental studies, paper chromatography again proves its worth. It helps in the analysis of pollutants in air, soil, and water. By identifying and quantifying these pollutants, Scientists can assess the impact on the environment and devise strategies to mitigate it. In the pharmaceutical industry too, this technique is widely used. It helps in the separation and identification of components in a drug. This is crucial to ensure the efficacy of the drug and to detect any impurities that could potentially cause adverse effects. As you can see, paper chromatography, despite its simplicity, has a myriad of applications. It's a testament to the fact that sometimes the simplest tools can be the most powerful. This technique continues to evolve, finding new applications and refining existing ones. Paper chromatography, a simple yet powerful tool, continues to be a cornerstone in scientific research and investigation. Subscribe for more because you won't find me later.